Okay, hello. Welcome, guys. This is going to be episode number 3 of my podcast. I am joined, and we've been on a call for a little bit, okay? But I haven't asked you on purpose how to pronounce your last name because I want to botch it first so that then
Please.
you can say it.
Yes, like you did with Alan. It would be
Yeah,
an honor.
exactly. You enjoy that quite a bit, so I thought I'd get
I
you with some
enjoyed it,
revenge to
yes.
start off. It's Steven Boeynaems.
Boeynaems, close.
Oh, close, close.
Very close.
Where's
It
the last
looks
name
more
from?
it's a Belgian name, so like a Flemish
Cool.
name. Flanders is like the north of Belgium, where we speak Dutch, just like in Holland.
Oh,
But for
cool.
some reason, we have the archaic spelling of our names, whereas in Holland they sort of updated their spelling. So our names look quite intimidating, but they're actually pretty easy.
I
I
believe
don't know
that.
why.
Alrighty. And per your Twitter page, you're a neurobiologist working on LAS, ALS
Yeah.
at Stanford.
Mhm.
But you're a lot more than that, and so I'd like to start with your own proper introduction because I don't think I could do it. I've visited your website, I've seen some of your work, your TEDx talk, and it's all very awesome. But yeah, why don't you tell us about yourself?
So yeah, like I do identify as a neurobiologist. I originally studied like bioscience engineering, so I do have an engineering background a bit. Got into genetics, started working on very basic fundamental biological questions in regard to evolution and so on. Through not getting a certain fellowship to pursue that work, I had to switch careers, and I really by accident ended
Who
up
denied you? Who denied you?
a funding agency in Belgium. But like
Okay.
I had to sort of pivot and switch careers, and I chose a more medical field, and I by pretty sheer luck ended in like a lab studying neurodegenerative disease, like an ALS lab, and yeah, then I sort of got really fascinated by what at that stage to me was just a very interesting biological problem. Why do people get ALS, this disease? And we can talk later on what ALS is exactly.
Hmm.
But then, sort of again, by yeah, sheer luck, with being at the right place at the right time, I started working together with the Belgium ALS patient organization. And so I started meeting a lot of ALS patients over the years. And that all of a sudden turned what for me was an interesting biological problem into more of like a personal goal where I wanted to
Cool.
dedicate my career to. And
That's
also,
amazing.
that made me move to the US as well to pursue more work here at Stanford. But besides ALS and other neurodegenerative diseases, some related dementias and other motor diseases, I have a broad interest in biology in general, and sort of still that the need to know of like how evolution works and how organisms adapt to environments and so on is still there. So my science definitely spans a bit more than just the brain. Like we're also collaboration working on plants, bacteria, and a ton of other funky organisms to like figure out really how biological systems work, adapt, and eventually, given my engineering background, getting ideas from nature to come up with novel therapeutic strategies for human diseases. So that is sort of
That's cool.
me, my professional me in a nutshell. And but obviously, I have a life outside of science. I like to stay artsy, busy, and yeah.
Yeah, that much is apparent just from your background, even the way that you dress
Yes.
somewhat. You don't dress like a scientist, right? So yeah, pretty cool.
If
We're
if
both
I got a
sharing beats there in the arm. So
if I got a nickel for every time I heard that, then living in the Bay Area on a postdoc salary would be definitely way more comfortable.
most definitely. All right. So education background, I guess then um engineering, could you be a little bit more like specific on that and
So it was definitely not like the traditional hardcore engineering was really bioscience engineering. Uh,
okay.
but that was like I really loved that education. It was like originally in Belgium, like let's say 10 years before I started those studies, it was still called agricultural engineering. It
Oh,
was just
cool.
only in sort of the last years then that more chemistry came in, genetics came in, and sort of then they expanded to bioscience engineering. But what was really fun is that our faculty was still sort of older faculty at the department were still sort of deep rooted in that historical setting.
Mm.
We are agricultural engineers. So the first 2 years, literally, we had to learn how to like grain is transported in like these machines, like these big machines that harvest the grain, and how that is transported with these screws and like
Like irrigation systems and
and
stuff,
like
I'm guessing.
stuff like that. And yeah,
Uh-huh.
then like after 2 years, you could start specializing, and I chose to move a bit away from really the mechanical engineering and so on, and move towards genetics because that was attractive to me. And yeah, sort of stuck with it. Because like if you would have asked me as a kid, I always knew I wanted to be a scientist, but I had always envisioned myself sitting somewhere in the rainforest looking for bugs
Mm.
would be my career. It's only in university that I sort of realized that actually you can explore the world as well in a lab. Because like sure, the frontier of science, you always think about space, you think about the deep ocean, the jungle, discovering new species, but actually just looking at our own cells in a petri dish, there is still so much we don't understand. And then by realizing that in university, I was like, okay, I just want to figure out how cells work. And then later on in life, I figured out I actually also really want to know why they don't work sometimes, and how we can, yeah, hopefully one day help come up with some drugs for diseases.
Awesome. Yeah, the universe, I guess it really goes inwards and outwards, right?
Yeah.
And there's a lot of I guess correlations to be made in your talk that I'm gonna reference from the TEDx talk that you did. For example, I like how you brought up an analogy of a traffic jam, for instance,
Yeah.
um, to kind of explain what the problem
Hmm.
of the what was causing the ALS or whatever. Like you'll
Yeah.
explain later because I am
Yeah.
I'm gonna botch it for sure. But yeah, I thought that was very interesting. I agree that we can look anywhere and find patterns and find correlates
Hmm.
and kind of like make sense of things, right?
Yeah. No, completely agree. I think also, like, in a way, like every scientist has sort of their personal style or is good at a certain thing. I honestly am not that great at like a specific, like super cool, fancy technique or so, but I think my strength is like looking for patterns and like
Mm.
trying to
You're an
connect.
explorer, aren't you?
Yeah, like I am a bit like I was always as a kid looking for bugs and fossils and so on. So you learn to recognize patterns, and I think I just do that in the scientific literature as well. Like when you're studying a brain disease, and all of a sudden you're gonna study a bacterium or a plant because you think you may learn actually something from the plants to like apply them later onto the human brain. So it's trying to come out like there's so many patterns in nature and in the world
Hmm.
around us, and I don't know. I think I just have an eye for that. And that is,
Yeah, I think maybe it's a little early in the conversation for me to attempt at this assessment, but like this is how I classify myself, and I see a lot of that reflected. So, would you say that you're kind of a jack of all trades and master of none in in a
yeah,
certain
definitely,
sense? Yeah,
definitely.
that's what
I
I am
do a
too.
ton of things.
Hmm.
Definitely not gonna claim I'm the expert at at any of them. There's definitely people way more qualified in all of these aspects. The thing is sort of what has been my strength is sort of having a type of knowledge of certain fields and be able to bring people together in the room.
Connect things.
Yeah, connect the dots and mostly connect people. Like
Yeah, that's what I love doing as well. So yeah,
yeah, no.
I can completely identify, and that's so awesome. So how did you end up at Stanford? What does that mean when it says Stanford on your profile?
Like again, it all started in Belgium. I was studying evolution, and then I didn't get a grant because in Europe it's sometimes difficult to get grants for really strong fundamental research. I pivoted my research to neurodegenerative disease, and then the disease that became a personal thing. And yeah, I literally met my postdoc advisor. Uh, I met him during my PhD in Belgium. He was an invited speaker of a conference we were organizing on ALS. And yeah, we started talking over a few beers and we had similar ideas and started collaborating and we just really hit it off. There was a really good personal connection. He had like the mentoring style I was really looking for. He was really supportive of my career from the start. And yeah, I just the moment I finished my PhD, I know there was one place I wanted to go, and that was his lab, and it turned out to be here at Stanford. So obviously, I didn't mind moving from rainy Belgium to sunny California.
How long
So
has it
yeah,
been now?
I've been here for around 3.5 years. So,
Very
and
cool.
yeah, it's been a great ride, and I definitely am not leaving California anytime soon, or the US for that matter. Europe
Hmm.
is calling, but like for now, it's like if I can visit, that's good. I still am not tired of my stay in the States.
That's awesome. Did you get your English in Belgium then? I guess most European countries are pretty good at
Yeah,
English.
yeah, we start getting English in like second year of high school. Yeah, yeah, we don't have the middle school system and so on. So, like, I think when we're like 14, 15, we start learning English.
All right. So you joined the lab then,
Yeah,
and then at this lab they already are working on researching ALS, and that is kind of how you are brought
yeah,
into it. Or you didn't have a prior interest then before the lab. It was more like
yeah.
your
So, yeah,
mentor
in
brought you.
Belgium during my PhD, I was working in ALS lab, and now that is why I
Mm.
specifically moved to this lab because they're
Oh,
also,
okay. So you met your mentor through working on ALS,
yeah,
or
yeah, yeah,
okay, cool, cool, cool,
yeah.
cool.
I
So
already
it was.
met him in Belgium, and we were doing accidentally we were doing very similar experiments, and
Mm.
I decided like, hey, we're doing actually the same thing. We came up with
Are
the
any
same.
of those experiments something that you could share that would make sense to me?
Oh yes. So yeah, yeah, no, 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 great. So the lab here at Stanford, specifically, they were pioneering based on like work from my bosses. His so my boss is Aaron Cutler, and he sort of pioneered the use of yeast, like baker's yeast, as a model to study neurodegenerative disease and most specifically ALS. And he started doing that already previously in like his mentor's lab, which was Sue Lindquist, who unfortunately passed away last year, who was at Whitehead Institute at MIT. And so you can think like, why would you study a brain disease in a single-celled fungus that doesn't have a brain?
Right.
For a long time, and things are we are in the mammalian field, like in the human cell field, are slowly catching up, but it will still take us some year. The yeast cell is definitely one of the cells on this earth that we know most about. We know its biology very intimately. They're slightly more simpler cells than some of the human cells we study. Like neurons are these extremely long cells. They're like difficult to work with. Yeast
Is
cells
it
are
is it like less components, or is it just the
they
structure?
have way less genes, and so because they had way less genes, they were one of the like I think they were the first eukaryote cell being sequenced. So we knew the yeast genome very early on, and also people started. There were a certain few genetic tricks that really worked well in yeast cells that don't really work as easy in human cells. So people started making these enormous collections, like libraries we call them, of yeast cells which would miss one gene. So this yeast cell lacks gene X, this yeast cell lacks gen Y, this gen Z, and so on.
Are those engineered by you guys, or are they naturally lacking?
No, so
Did you find them?
people have engineered them, and the yeast field is also like it's a field that always realized the value of sharing technology. And so there were certain labs in the yeast field that early on generated those libraries and freely sort of distributed those. So a lot of people could work with them. And so the thing what you can then start doing is, okay, you can express a toxic protein that is found in the brains of, like, for example, ALS patients. And these proteins, and we can come back to this later, what they do is they clump together. So in all of the neurodegenerative diseases, pretty much like Alzheimer's, Parkinson's, ALS, some dementias, you get like these clumps of proteins in your brain. And that's toxic
Yep.
to human cells. Now, the key insight was if you express that human protein in a yeast cell, the yeast cell gets sick as well,
Oh wow.
like a neuron would. And so the nice thing about that system is now all of a sudden you could express that protein in every single yeast cell that was always lacking one gene.
Cool.
And what you then do is, like, in a high throughput fashion with robots and whatever, uh, you could start looking at these cells, how they deal with it. So most yeast cells will just be very sick. But sometimes it would happen that you knock out gene X and the yeast cell would even be more sick. Sometimes
Oh,
you would knock out gene Y, and all of a sudden the yeast cell would grow better. It would be resistant to the toxicity. And so this is what we call genome-wide screens. And so yeast was really like one of the early, like it's the only eukaryote at first that we could do that in, and that is why what this lab at Stanford was doing.
sweet.
And so they were screening the entire yeast genome to see which genes modified the toxicity of the pathway. And now,
To
since
then isolate the problems and then know
yes,
which genes to play around with, right?
yes, and since like yeast has like a lot of genes conserved with humans, we could then look. Oh, is the ortholog of this like the human version of this gene? Is it also implicated in ALS,
Hmm.
Parkinson's patients, and so on? And so I like the Stanford lab was doing that already for years, and I initially got trained in a yeast genetics lab where I was studying just yeast evolution. And sort of by not getting that grant, I
Oh, cool.
did to neuroscience. Uh, and then I realized, hey, I can still look. There's people in the U.S. using yeast to study neuroscience. Maybe you can still do that as well. And
That's so cool.
I started working on a project, and then actually like the person that sort of inspired me to do work like this, my boss, and like he was an invited speaker at a conference. We met over drinks, and we were like, hey, we should do this together. And yeah, that's where we connected. And yeah, it was there was definitely some scientific chemistry there, and I was immediately like, yeah, we should do this.
That's amazing.
Just know it
All
when you
right,
meet a
and then
person, like if it clicks or if it doesn't, and the same is in science, right?
yeah, of course. Yeah,
So yeah,
and then and so you've been working on say like mapping the genomes of these yeast cells and figuring out like where to look essentially, and then
yeah.
I think you moved on to something like flies, right, or fruit flies, or is that
Yes.
a or am I?
So then we started collaborating with Stanford. In Stanford, they had all the robots in place, all the yeast libraries. So we decided that they would do the yeast work, and that
Cool.
I would in Belgium focus on trying to do the same effort in fruit flies. Fruit flies, Drosophila, also very well genetically characterized. Also, you have these large collections. Only thing is, it's hard to do them like the whole genome. So we did what we did is we first let the people at Stanford finish the yeast screen and all
Hmm.
their hits, so the genes that modified the toxicity. We then looked for the fly versions of those genes and tested those in a fruit fly model. And sort of then we combined those efforts in two papers. And like it was really cool because we both like through both yeast models and then fly models, we really singled out on the same molecular pathway. And then like there at the same time there were two other labs in the US that had taken different approaches and they also ended up on that pathway. And so that's
That's amazing. Is
still
there a
like
step up from flies like rats or something like that, or is that too
yeah.
large an
So
organism?
the like there's multiple organisms that then have like or closer to human, if you will. You can go to
Pigs are
zebra.
pretty close, I think, right?
Yeah, yeah, yeah. Oh, that's super close. But those are very labor intensive to work with. So
Mm.
most people would then like go for mice. There's also a lot of people working with zebrafish there because they're a bit a faster model, but like it's a fish.
What does that mean, a faster model?
A faster model means like in at least if you work with fish embryos, uh, your experiment won't take that long. Like a mouse will
Mm.
live around a normal healthy mouse will live in a lab around 2 years maybe.
Mm.
Now the ALS mouse model, so you can also just like you did with the yeast, overexpress like a toxic human protein in these mice, and they will develop a disease that looks like ALS. So, but then these mice still will live for 100 days. So if you want to start testing drugs and so on, that takes a lot of time.
Right.
In a zebrafish embryo, you can just take the fertilized egg cell, you inject an RNA coding for the toxic protein, and then the zebrafish will develop. And so if you inject that toxic protein, you will the developing zebrafish will get problems with their motor system, so they won't be
Mm.
able to swim properly and so on. And that's something you can see in a day or two. And
Cool.
a day or
Okay.
two is different than 100 days.
100 days.
Now,
Yeah, that's
obviously, a mouse is closer to humans, so I think like these bottom-up approaches, starting with very simple models and building up to models which are closer and closer to humans, I think it's definitely a valuable approach still, and that's why also so many people are still doing that type of research. And labs often combine a few steps in that like cascade towards the human system.
Yeah, I think this is very consistent with the start of the conversation, right? There are repeating patterns, and yeah,
Yeah,
organisms
no,
as simple as a cell or a human
yes,
being, they'll share. Yeah, they'll
no.
share a lot. So
And like I like also that you say repeating patterns, because the work I was doing on evolution in yeast was actually looking at junk DNA and if it plays a role in evolution, can play a role in evolution or not. And specifically, I was looking at tandem repeats. So small like words in your DNA code that are repeated
cool.
head to tail. And I had to switch from evolution research to something more applied to get funding. And exactly that year, I had to switch. The major genetic cause of ALS was found. It was a mutation people had been looking for years, and it turned out to be a repeat expansion. So again, like these words repeated. So I started
That's cool.
studying repeats in yeast, looking at sequences and finding the repeats. And then in ALS, all of a sudden a repeat pops up. So I had to
Shit.
do it. And
Well, we should
repeats
talk
have,
first for anyone that doesn't know who what
yeah,
ALS is, right?
ALS.
Like, yeah,
No, that's
let's
an
start with that.
epitome.
I will give my shitty definition of ALS, but it's basically your motor skills deteriorate. You could say, I guess, deteriorate to where
Yeah,
you become you can't move, right? Like you can't move, but you're still conscious,
yeah,
and you can't move basically any muscles in the worst parts of it. You could argue.
yeah. So what happens with ALS? It's like ALS stands for like amyotrophic lateral sclerosis, and it's pretty much a complicated name. That means that there are specific nerve cells that connect your brain to your body's musculature, like your biceps, triceps, whatever. And it's those cells that die, the ones that. So it like I often compare it to like when I give like lectures to kids and so on, I compare it to like your brain. It's like your computer. Your computer will like power a robot or like will
Hmm.
give commands to a robot to move. And so the computer needs to send like a signal to the robot. And like in a machine, that's through an electrical wire. Well, exactly these nerve cells are the electrical wires. And when you, for example, have a car crash or like you break your spine, like you rip your spine or whatever, then you just rip the cable. So what
Right.
happens in like these diseases, the neurodegenerative diseases, you can compare it to like over time the cable slowly rusting and
Mm.
not conducting anymore, and eventually it will also break. And so that is what is literally happening. So a lot of people think ALS is a muscle disease because these patients present with muscle wasting, so they will lose muscle mass and they will start to paralyze. But it's actually a problem of the brain and the spinal cord. So it's really the cells that send a signal to the muscles that will cause the paralysis. And
That's okay.
yeah, like Americans often know it as Lou Gehrig's disease after like this a famous baseball player from a few
I see. I
decades ago, I
see.
guess. And like people still sort
Stephen
of
Hawking had this too, right?
yeah, Stephen
ALS.
Hawking had this. Yeah, and like people still may remember like the ALS ice bucket challenge. That was
Yeah, that was huge. I
yeah,
always knew about the ice bucket challenge. I never knew what ALS was still though. So
yeah, no,
half a
so
success, I guess, on that front. Yeah.
yeah, no, definitely, yeah, because like ALS was definitely not a well-known disease, but sort of after the ice bucket challenge, it sort of yeah got
Definitely helps.
the attention it definitely deserved.
That's good, and that comes with attention. Comes with like I suppose grants and money for people
Yeah,
researching and for the research
no,
in itself. So
yeah,
that's a good thing because it's a scary prospect. And yeah, I don't want
it
to
is.
I don't want to like I don't like personally. I've been feeling these things that make me like a little bit paranoid, right? Where like my muscles, they're not like sometimes I don't. I get like jolts. Like it's like I feel like my muscles contracting
Oh yeah,
at
or like
many points during the day actually, and this is happening like kind of daily. And I
yeah,
was like, oh
like
shit.
do you also sometimes get the feeling that sort of like a muscle will start like twitching?
Yes,
Like
yes, a lot.
those are fasciculations. Yeah,
That's
those
not good, is it?
those things are normal. Everybody has them. Like often, like people also like specifically if you're tired, so you can get that with your
Yeah.
eyelids as well that they start twitching. I
Mhm.
also have my arm, like sometimes like certain muscles that do that often. It's a very normal thing. The
Hmm.
problem is if you work in an ALS lab and people that study ALS, they're all paranoid.
Right. Yeah.
So
Right.
every time they feel a muscle acting up, they're like, oh my God, I gave myself ALS.
Yeah, I shouldn't have
So
given.
yeah, people studying ALS, a bunch of them are like, yeah, overthink these things. But yeah,
That's incredible. Yeah, I've informed myself too much where I'm like, oh shit, whenever my muscles
it,
twitch. But
it's
it's happening
definitely,
often, and it's happening more than I remember before. It could be
yeah,
confirmation bias, but I
yeah.
think it's not.
First of all, definitely confirmation bias, and also like these things tend to like also relate to exercise, like if you're
Yes.
sleeping well, whatever,
Hydration,
well, hydration. Like there's so many things.
stress.
The
Yeah.
problem is like it's a lot of these like diseases in like very early stages. Like the symptoms are super vague, right? It's only
Hmm.
like in late stages of disease that you really see like symptoms that are like, okay, this points at a disease. Whereas like symptoms you get early on are like, so many people have this.
Yeah, I've been worried. I
There's
was worried about my
fasciculation.
circulation, but now I see that I need to be more worried about my electricity, right? Like my
Yeah, I think plenty of sleep, good diet, and some exercise will work perfect.
All right. When does someone then start getting worried? When is it? When do you actually find a patient that you're like, okay, you're on the way to ALS?
So the thing is, I also don't see patients myself. So
Hmm.
I'm a
True.
PhD and I'm an MD, so I definitely can't give diagnoses. But generally, most what I hear from my MD friends, generally most people will present to the to their GP at first with some weakness in a specific limb or
Hmm.
difficulties speaking or swallowing, depending on where the onset of the disease is. And so then ALS is like a bit of a tricky disease to diagnose. It's like the diagnosis happens through elimination, because specifically in the early disease, like for example, your calves, some weakness in your calves, and not being able to reach the upper shelf in your kitchen, or like dropping things because your hand you lost
Your
some
grip.
like
Oh wow.
grip strength there. That could be a ton of things, and so what happens is like a neurologist generally will start ruling out thing after thing, and like if there is progression and so on, and all other options, more benign options, are ruled out, then often that leads to a diagnosis of ALS. But again, ALS is also on a spectrum of diseases, and it's becoming more and more clear that it's actually a very complicated spectrum, where
What
people.
does that mean? What does that mean?
So we used to think, for example, of ALS is
Binary.
one disease, and you have these motor defects, and that is it, and the
Hmm.
rest you're perfectly normal. Now it turns out that there is a type of dementia, which is not Alzheimer's, but we call it frontotemporal dementia. Frontotemporal just refers to the brain region, the frontal region and temporal region, where the degeneration hits first.
Hmm.
So it's something very different, right? These people have like eventually memory loss, but they initially present with behavioral changes, problem with speech like aphasia. So like
Hmm.
being able to speak with their mouth and tongue, but sort of not being able to come up on a word. So like they would see a picture of a cow and they know it's a cow, but they just sort of cannot find the word cow to say.
Right.
Something like very simple explanation of what aphasia could be.
Hmm.
And sort of something completely different than ALS. But over the years, it became clear that there's often ALS patients that have some like more behavioral problems as well, and there's often like these patients with this type of dementia that would also get mild motor impairment. And then
Hmm.
there's patients which have both of the diseases. And so it turns out then looking at the genetics and looking at the pathology, so what is really going on in the brain, um, there's often overlap. So the mutation in the same gene can give both of the diseases or a mixture of them. And so then it became clear that ALS patients are on the spectrum with this dementia, and they can have pure ALS, or they can have just pure the dementia, or they can have a mixture and everything in between. So you really have patients that, all patients you can put on that spectrum. Now that is only with this one dementia. there's evidence that certain mutations that can give you ALS can also give you a bone disease, or really like a
Hmm.
muscle disease. And sort of there's more and more genetic overlap between a variety of these conditions. We were also involved in like the identification of a disease gene that now also is a risk factor of ALS, that actually has, from since the 90s or so, it has been implicated in another disease, which is an ataxia, which mostly hits like the cerebellum, like your small brains. Like,
Hmm.
and sort of, again, very different disease than ALS, but there seems to be some genetic overlap. And sometimes, in rare cases, you have families where the disease runs in the family, and one family member will be diagnosed with really degeneration of the
Hmm.
cerebellum and ataxia and all of the associated symptoms. And one patient, like one other family member, presents with like a pure form of ALS, like a clinical pure form. So you couldn't distinguish it from another ALS patient. So there is clearly overlap between all of these diseases. So again, it will take a few like more years for that to be truly accepted, because but like there is
Yeah,
more and more like acceptance of the idea that diseases aren't limited to like, okay, this is a disease, this is a disease, but
we're
there's
always trending
like
towards that,
this
right? And on,
blurry spectrum,
yeah, I think
which
that's on all
again,
fronts.
yes, and again, which is like an opportunity, right? Because not like so much, like the field of neurobiology has been studying, specifically neurodegeneration, has been studying all of the diseases for sometimes decades. So now, if we know, oh, ALS is linked to this ataxia, or ALS is linked to this dementia, we now all of a sudden have access to this wealth of information on this other disease that we can look into, and maybe, hey, maybe this works for actually ALS patients as well. And we can again start finding patterns and connecting the dots.
Hmm.
So
Now,
I
now,
think,
go on.
go ahead.
I was gonna say now, like when you get older, for example, by
Yeah,
default you're in a state of decomposition, I guess, to some degree. Yeah, to put it
we're all
lightly.
decaying.
So like in a sense, does everyone have ALS? I mean, old people start to forget things. Old people move slower, and I know that there's other factors, obviously, but there has to be some of that rust accumulating, regardless of if it's accumulated too
Yes.
early on, or right.
Yes. And so that depends from disease to disease. So ALS is a bit peculiar in that. So I will come back to ALS, but let me start first on Alzheimer's disease.
Hmm.
So we see that the older you get, the higher the chance you will get Alzheimer's. And eventually, even in old people that passed away because of other reasons, if we perform autopsies, we sometimes see that they actually have the pathology of Alzheimer's. They just didn't get the symptoms yet. That could mean that they were still developing the symptoms, or that somehow their genetics made them resistant to it, which is already
Mm.
an interesting idea. So, and from Alzheimer's, we know very well that there are some people that have mutations that make them resistant to developing Alzheimer's disease. And there may be genetic difference in genetic makeup or lifestyle that make you, yeah, more prone or less prone to develop disease. So, but that is the important thing. Like Alzheimer's, if you get old enough, like your chance of developing disease is getting higher and higher. ALS, something weird is happening. Most people that get ALS get it in their fourth or fifth decade. So when they're in their 40s, 50s. Uh, and then if you look at older people, their risk of developing ALS is
Mm.
going down.
Okay.
And so the thing is, we don't have a good answer why that is happening. Is that there's two options. Like for me, like neurologists may or may not agree with me, and everybody has probably different opinions. But for me, it boils down to two things. Or there is something really about sort of that early aging around like 40s, 50s, and so on, that makes you specifically vulnerable for ALS, and certain processes that would make you more vulnerable go down in late age. That is a possibility. Another possibility is that ALS is generally a very aggressive disease, and all of these neurodegenerative diseases, they actually seem, their onset really seems to be happening probably in people their late 20s or 30s maybe. and only over decades of aging then they really develop the disease. And so, like for me, it's also likely that since ALS is a very aggressive disease compared to other neurodegenerative conditions, for example, an ALS patient that comes to the clinic and can present with, for example, a bit of weakness in the calf or in the hand grip, uh, sometimes they pass away after 2 years already, or half a year even. Like the progression of like just having somewhere a bit of weakness in one of the limbs to going to a wheelchair to passing away is sometimes as short as 2 years. Some people live longer with it for sure, but often it goes very fast, which to me suggests it's a very aggressive disease. So I
Mhm.
think that sort of that we see most patients getting it in their 40s, 50s is maybe due that it's just a very aggressive disease. And the chances of like then presenting with it truly in your 80s, 90s is just very low because probably you would have already passed away because it's such an aggressive disease. So yeah, there's two possibilities why that is, but we don't really know. But definitely aging and neurodegeneration is an inherent process that all of us go through. It just, again, it's a spectrum, right? When do you draw the line of like everybody that gets older, like has some memory issues eventually? There's some people again that have perfect memory when they're old, but most people will sort of like talk to your grandparents or whatever, like they sort of like forget things easier and so on. So it's an inherent process of like human aging.
Yeah,
And it's only like where do you draw the distinction really? Okay, this
this
is
is too much,
disease
too
and
much
this
too
is
early.
still normal aging. And it's a spectrum, right? And
Hmm.
there's no clear definition of like this is disease, this is not disease.
Yeah, we have to arbitrarily pick a point and this is say
Yeah.
this is too much. Yeah.
And so, but definitely the age related degeneration is key to all of these diseases. So if you really want to find a cure all, or at least like a way to slow down most of these diseases, yeah, you need to just study aging and what aging really is and why we age and how we age and if we can revert it or slow it down. And that is, I think, where a lot of the future of the field lies as well.
Ah, are you into that? Slowing it down, reverting it?
Okay, I live in Silicon Valley, but like I don't want to live forever. Like there's like this obsession with age research here and so on and by these things,
Yeah,
which
because
is,
yeah,
but
it's
I think we have to stop thinking about lifespan
mm-hmm.
and start thinking about health span. So instead of thinking like, oh, I want to live till 100, uh, yeah, I honestly don't want to live till 100 if that means that from my 80s on, I'll, yeah, be sitting in a wheelchair or whatever, or like can hardly move around, or people have
Mm-hmm.
to take care of me like for 2 more decades. I honestly wouldn't want that. If I can live until I'm 80 and have functionality and feel great and so on, and feel healthy and don't have memory loss and whatsoever, then I have a longer health span. And I think that is more important than lifespan. Because like we also start seeing like, sure, we have increased lifespan dramatically in the Western world, but that is also, and specifically because birth rates are decreasing, that is causing also quite some socioeconomic issues, right?
Mm-hmm.
Because like, specifically coming from Europe, from a welfare state where people get a lot of pensions, where like everything is well supported, and I really am happy for that, and I'm proud of that as a European. But the problem is the system becomes unaffordable because people keep on living and living longer,
I see, and they're
and
not producing.
they're not producing, and the young kids have to pay for that. But like it's like this vicious circle, right?
Hmm.
Specifically, if birth rate is dropping, like you can't keep up with that system.
Right, right.
So we
Yeah,
need to make people not just live longer, but live healthier lives, and like
so that
make,
they can continue to do things and yeah,
yeah, not
live their
necessarily.
lives.
I'm not arguing that everybody needs to keep working till they're 80, right? But just that some of the costs of having to care for a dramatically expanding older population on society will decrease. And so that's why I think starting to think about aging research as increasing health span rather than this obsession with lifespan
Hmm.
is a good move because
Yeah,
it will like solve a lot of issues.
I like that. There's also, I mean, this is somewhat adjacent, but I think in Eastern cultures, like elderly people aren't seen as they are in the US. Whereas elderly people in the US, they're kind of like, okay, just put them in the house for old
Yeah,
people, and they're not making any sense. And whereas in the East, it's more like they will make the most sense because those
yeah,
are the people that have lived. So there's a utility, even if they're not actively working the field or some shit, they're
of
still
course.
contributing to the culture in a way,
No,
and you know,
yeah,
their knowledge, their wisdom is respected. It's
yeah,
craved upon. So yeah, I think that there are other things, right, like disconnected from science that we could do to,
no,
yeah.
definitely, definitely, for sure. And yeah, and also it's like, again, a bit unrelated, but it just made me think about like how we view the elderly and how we also deal with these type of issues of like if you have to indeed care for somebody who has dementia. Uh, somebody who has dementia will like lose sort of short-term memory, and a bit like they often have like still very deep-rooted memories from their youth or so, or when they grew up or an important stage of their life. And so in Holland, they have this amazing facility. So it's like this elderly care facility really focused on dementia patients. And so what they did is they created sort of this mini village, and every house, a few patients live, but every house is decorated in the style of the decade they still
No way.
best.
That's so
So some
cool.
people live there in the 50s, some people live there in the 60s, in the 70s,
What?
and sort of they live there together. And there's this small village square and so on. And like, yeah, their kids and so can come and visit, but these people are happier because they sort of they don't wake up every day in a modern world
And
and
they're
be,
confused, yeah.
where am I? Because they sort of have somewhere these memories still from earlier on in their life,
Hmm.
and it's and like seeing people with dementia, then you sometimes they don't recognize you anymore, or they're like, where am I? And they
Hmm.
start recollecting things about their past, and so them living in a setting that reminds them of their past makes them way more calmer, and like makes them more happy because they feel at home rather than feeling like waking up every day in a place you don't recognize. And
That's
so I
fucking
think
crazy.
like
Yeah.
those type of efforts are like specifically for dementia and disease related care. Like I think those are definitely the future.
That is so cool. Yeah, it's pretty crazy to think about, like, who is the person that wakes up, right? If you don't have those memories, if you don't have those
Oh,
but because you have some parts of your brain that are working, and that you know, like, you know words, and you know
yeah.
meaning, and you understand the cultural context on whenever you lost
Hmm.
your thread, right, towards today, I guess.
Yeah.
But but then you don't remember certain things like people, or and that's,
Yeah.
yeah, that's.
And that is specifically also tough for the people involved, right? And
Mhm.
also like and to sort of get back on the ALS part of the thing, like the the ALS patient is obviously going through something very very traumatizing, but like the family members surrounding them, it's yeah, seeing like specifically since ALS is a disease that hits you in your 40s 50s. That's why it also like getting to know the patients and their families, it resonated so well with me because the patients were sort of my parents age
Mm.
and the kids were often my age, and so I remember going to like uh like these retreats for patients and give talks there about my science and so on. And then people afterwards walking up to me, people my parents' age, like, "Hey, you remind us of our son," and oh, thank you, this, and like sort of that made it like all of a sudden very clear because like their kids were like having to take care of like yeah their parent that is like really slow, like not slowly but like very fast, like progressing over 2 3 years, like from being an active an active 40-year-old to yeah somebody in a wheelchair and completely locked in, not able to communicate and so, and that is like yeah very very traumatizing, and that's why also ALS stuck so much with me because it was relatable. Like I
Yeah,
was seeing these patients and I was like these could have been my parents,
yeah.
and like and it was sort of like damn, if I can contribute here then yeah I would love to.
Yeah, from what you've told me, it sounds like everything kind of pushed you in the same direction from different angles, right? Like
No,
your
like some
curiosity for
sometimes
biology and
I don't necessarily believe in like a higher purpose or like a path or like fate, but like the universe, like life throws like curveballs at you, and you just like you go with the flow, and
yeah.
you see where you end up, and you pick up on signals, or like, I think I'm on a good track, and this is gonna
Hmm. Yeah.
give my life meaning, and so and a sense of,
Exactly. I mean, you get rewarded,
and
right? I mean,
yeah,
it's yeah. You don't have you can read it as mystical as you want, but the fact
yeah,
is that you liked biology, and then you liked people, and you felt more connected to them, and obviously, if you were to do something else at that point in time, then you would
yeah,
probably wake up feeling like shit because you didn't do the thing that you were
yeah.
invested in. That's just
No,
how life works.
no,
So
completely agree.
let's play this video. This
Oh,
one is
awesome.
the mannequin challenge,
Yes.
and yeah, I found it, and then asked you to play it. It's pretty cool. It's pretty well produced. This is the Belgium lab.
Yes, this was still in my lab in Belgium.
There you are
So,
with the sideburns. Can we pause on the sideburns really
oh
quick?
yeah, when I still had was rocking my sideburns.
There it
It
is. If you
was
could see.
glorious.
Um,
So yeah, this was I think the year after the Ice Bucket Challenge, and the Belgian patient organization wanted to do again something for ALS. And so after the Ice Bucket Challenge, like these type of challenges on social media became also more and more prevalent. And so around the same time, the year later, somebody started the mannequin challenge. And since and you'll see it at the end of the movie, like every day is a mannequin challenge for an ALS patient, because if people remember the mannequin challenge, you had to like sort of like put
is that an
yourself.
actual ALS patient?
Yes, it's an actual ALS patient, and
Hmm.
like yeah, they're every day is a mannequin challenge for them, because they're like, yeah, they're paralyzed.
Yeah, I
And
see that his eyesight even is fixed, right? They can't even move
yeah,
the eyeballs.
so generally they can move their eyes. So it's super weird. So like in ALS, like eventually most of your muscles can paralyze, but it's only the voluntary muscles. So your heart muscle and the muscles of your GI tract, uh, those still work, but all other skeletal muscles, we call them, so the things that make your body move, they tend to paralyze. The one exception to it, the muscles that move your eyeball. So if you remember, like initially, like Stephen Hawking, he had like this PC, and they had like this camera pointing at his eyeballs. And by then looking at a keyboard
Mm,
screen, you could actually
cool.
type an email or so. Later on, that also became too difficult, and so on. And like also
Mm,
first, like also like a sort of a sensor here that he could twitch this muscle, and then could like also control his wheelchair and so on. But like a lot of ALS patients in later stages, they sometimes use this eye tracking software. It's remarkable how some patients, how fast they can type an email, and it's also like it's that's the
that's
thing
cool.
with like about ALS patients. Like there's a lot of dreadful diseases, and like ALS is sort of like you're really getting locked in your own body, and like your mind is still fully functional, fully capable, but you're sort of locked in, and it's like it must be insanely traumatizing. But what every ALS patient I met over the years, they're always so hopeful and grateful for all the care they get from their neurologist, from the nurses, from their family around them, and and they still find so much joy in like these smaller things. And I think that is like a lesson for all of us. Like I still remember like the first time I was talking to a patient, and he was sort of bragging about how fast he could send me an email with
Nice.
the eye tracking software, or a laser on his head, and like just pointing at the screen, and like they find joy in that, and like they're like grateful to still enjoy every day with their family, and find support in there. And that is like, yeah, that just, yeah, makes me
That
tear up
is
afterwards always, because like it's like our lives, we have so much, and then we still like often feel shitty, and then like talk to somebody that's going through something as traumatic as that, and then see that they, in the small little things and just connecting with family and friends, can still find so much joy, then it sort of puts your own life in perspective, and the small bumps in all of our paths.
Yeah, and that's kind of the thing, right? It's almost strange because it feels like no matter where you are in life, it's that roller coaster. It's up and down, right? And like, yeah, like no one wants or craves something like ALS to happen to them.
No.
And yet when you are there, it sounds as though as if they also just find joy and they can be grateful and they can live a life that is worth living from their perspective, right?
Yes. Yes.
And that's yeah, I think that's really great. And it's great to get the perspective, because then you can kind of be grateful of what you do have in the present moment. And
No. Yeah, I think that is always like, it's always good to sort of evaluate our own lives and see like where we're at, and like be mindful of things, be grateful about the luck we have, and like, but like connecting with people that obviously have it worse is I think always like a humbling experience, and sort of like, wow, I have to like put my own things into context and my own issues into context, and like just, yeah, soldier on, and like keep on helping wherever we can.
most definitely, that's incredible. Now you're also an artist. I'm gonna bring up some of your art now.
Wanna be artist.
Wannabe artist, you finished second in the contest. You're underselling here. All right, so let's bring this one up. So what are these guys?
So yes, this is some of my science. Like, you can see some of my paintings behind me. That is just abstract paintings. They don't really represent anything. But this is like some of my science art. And so what we play around here with is what we call liquid-liquid phase separation. And liquid-liquid phase
Lick.
separation is a complicated name for making salad dressing. Take your balsamic vinegar, take olive oil, put them together, shake them up, and you will get oil droplets dispersed in sort of your vinegar. And if you wait long enough, eventually they'll completely separate. So that
Mhm.
is what liquid-liquid phase separation is: two liquids that don't mix together. And so,
But if you wait long enough, they mix. What about water and oil? Would they
no, they
eventually?
completely separate. Sorry.
Oh, okay, okay, yeah, yeah.
Sorry. So they don't
Cool.
mix, and you can sort of disperse the droplets, but they never will fully mix. And so, why am I interested in salad dressing? Well, it turns out that we briefly touched upon this, but in like these neurodegenerative diseases, there's like clumps of protein in the brain, and normal people don't have them. And if you have Alzheimer's, you have specific clumps. If you have ALS, you have specific clumps, Parkinson's, and so on. So on. And so a lot of these protein, normally they are just floating around happily in the cell. And what we see in these diseases is that all of a sudden they become like rock solid, and they like freeze up and they sit there. And so if you think about it from a physical point of view, you can think about the proteins happily floating around. They're sort of behaving like a gas, like water vapor in the air.
Hmm.
And these rocks are more like snowflakes of our ice cubes. And so we never really well understood why this is necessarily happening, and if there's a direct switch from this gas-like to this solid-like state, or if there is an intermediate. Because like if you think about water, a snowflake will often like nucleate from a water droplet. So water can from condense from vapor to water as a liquid. Now it turns out that proteins do this as well in our cells. And sort of there is a bunch of like small compartments in our cells that are actually like phase separated, we call it, liquids. So what you have is normally these proteins float around, and sometimes they can like also condense into like a protein droplet, just as oil molecules would like always want to condense in an oil droplet. And sort of that liquid-liquid phase separation, so the demixing of these liquids, is also happening in our cells with proteins. And some of the proteins doing that behavior are exactly the proteins that then sort of freeze up and aggregate, like clump in our brain. So we and a bunch of other people like have been really focusing on that and how those processes could be implicated in neurogeneration. Now, if I explain it, it like sounds very physically and like boring, but I wanted to like come up with a more like brighter way of telling this story, which is where these pictures come in. So
Hmm.
what I do here is I recreate sort of liquid phase separation in the lab. So what I do, I take this like big petri dish, I pour mineral oil in it and pour then solutions of different proteins in there, and with different dyes I just found in the lab, and like food
Cool.
coloring dyes, and try to create sort of some people see like space in it.
Hmm.
uh,
Sure.
I always joke that I tried to recreate an artistic version of like the insides of our cells, like having all of these protein droplets floating around, because these
Oh, I
things are
love this one. This
also
one's
made
so
with,
cool.
they have egg white in there, which is obviously a lot of protein. They have milk powder in there, which is obviously also protein, and sort of, yeah, I this is my artistic representation of the inside of our cells.
That's incredible.
It provides something tangible about you think that proteins can form like droplets and can sort of bump into each other, because these are, I don't have videos here, but like these droplets will float around and like they bump into each other and then the colors mix. And
Oh, that's so cool. So these are
if
not
you wait
stills.
if you wait long enough, eventually everything will turn brown. That is what every art.
Everything just one
Too
single
many colors,
bubble.
it will get brown. So eventually, if you wait, because these things are ever dynamic, and like they always, these are really snapshots of like a moment, and sometimes you see them just fusing and the colors mix, and
Oh,
you
that's
could,
yeah. These guys for
it's,
here, right here, they're about to go
yes,
into the yeah.
yeah, and
You can
so
tell.
like, and that's what I like about it, because it also the fact, and I should have made proper movies of it, but I still need to buy a better camera for that.
Mhm.
Like that's what I like about the thing, because I can't freeze this process. It's there and it's happening, and you snap a picture, and that was like that moment sort of will never come back. And that's exactly the same in our cells. These things move around continuously, and they are so just so dynamic and ever evolving. And that's what
But
I like
that this
playing around with
moment will
this.
never come back. It doesn't sound too hopeful for ALS, right? Like if the aggregates, right? Like it's like
You
how
can
do you
you can,
separate them again? Because that
oh
sounds
yeah.
like the thing
So
to do.
you can reverse certain things,
Okay, okay, cool.
and like
So
in
the
certain
moments can come back then.
sort of moments come, well, you just revert to a previous state, but you can just, you have to go another way often there to get.
Okay.
So if you get, so the thing is, these protein aggregates form continuously in all of our cells. Like all of us are getting protein aggregates in our cells as we speak.
Hmm.
Only thing is, if you're young and healthy and don't have underlying mutations, uh, then your cells will just eat up these aggregates.
Oh, cool. Okay.
They will clear them, like they'll just throw them in the trash and they degrade them. The only thing is, if you have mutations in that system or overaging, that degradation system also like becomes more vulnerable, more less efficient. I mean, then you start building up these proteins, because we continuously, proteins misfold and aggregate in our cells, and but if you're healthy, you'll get rid of them. If you're aged, it's harder to get rid of them. And
Mm.
if you have mutations or certain environmental risk factors or whatever, then the buildup may at some point exceed a threshold, which will like prime you to develop the disease. But that is again then a thing that is hopeful, right? Because specifically, I just touched briefly upon it, but what we now know is that a lot of these neurodegenerative diseases, probably this buildup of these proteins and this aggregation starts happening early on in life, in your 20s, 30s, maybe, and only will represent decades later with the actual symptoms, the disease. It's difficult to study that, because like you would need to study people that don't have the disease yet but will develop it in a few
Right.
decades. So that makes it difficult. But the thing that makes it hopeful is like these people are slowly on their path towards disease, but they still live pretty healthy for a few decades. So there is so much room for intervention there. And now most of the trials are obviously based on people that are sick, and we try to slow it down. And specifically, if you have a fast moving disease space, let's say ALS, prognosis from getting the diagnosis till death can be like 2 to 3 years. Try to delay that is difficult. If you try to delay a process that is taking 3 4 decades, you have a lot more room to, like, if you delay it a bit slowly, you can maybe give a patient another decade of healthy life. And sort of the realization that a lot of these diseases, even though age related, start earlier on in life, but we can cope with it, is also hopeful, because there's the therapeutic window becomes like way wider. And if we can start treating people early on in life, then we probably can like really help people out in the long run, like really extend survival and like help the healthy lives of these patients, rather than just waiting till, oh, you clearly have this disease and you'll have only that many years to live, and now we're going to try to make that a bit longer. No, we have probably a few decades to try to do that.
That's pretty cool. That's pretty awesome. All right. So, I mean, this droplets to me, they catch my attention in the TED X talk that you did. I remember that you were talking about how, like, the gassy versus liquid versus solid nature of
Yeah,
things, and the solid's like the thing that no one ever wants, right? Like, it's
yeah,
in most phases of life. I mean, if things stop moving, then that's bad,
yes.
right? And
So
most of
yeah,
anything
yeah.
outside. So
Again, it's always nuanced, and you should definitely talk to Alan about this. There are conditions where solid is good,
oh,
and
really? Cool.
yeah, there are, but again, it depends from situation to situation. Context is everything.
Hmm.
So some proteins need to be solid for their function, but some proteins need to be gas-like for their function, and some proteins need to be liquid for their function, and some proteins need to
Cool.
switch between all of them. And it's mostly when really misregulated then it becomes a problem. If a
Okay.
protein that shouldn't be solid becomes solid, that's a problem. If a protein that shouldn't be liquid becomes liquid, it's as much of a problem. And so it's the cells always need to, these proteins evolve to do what they do, and the cell needs to sort of chaperone them and keep them in check, make sure that everybody is well behaved. And
I
it's sort
see.
of, definitely don't want to say that solid is always bad, because then
Cool.
Alan is
Cool.
super bad at me.
Fair, fair. All right. So solid is not always bad then. But what is it about it, the ALS, that makes
Yeah,
the transition to solid bad in this case then?
yeah. In
It
ALS
creates the
specifically, for
jams, right? Like the
yes, yeah. In ALS specifically, these proteins turning into a solid, so like literally freezing, like freezing up, as if you will, like an ice cube, that is bad, and we know that is bad. And what is the worst thing actually about it is that these things are not infectious, as in from person to person, uh, but they are infections on the cellular level. So they can, like, if you have like a protein aggregated and formed like this small like solid,
Mhm.
it can break up in pieces and can like jump to a the cell nearby. And if the cell nearby takes that up, like the same as like an ice crystal will form often on a nucleus. Like it will first need to nucleate, and it sometimes does that on a dust particle or like
Mhm.
mini ice cube, and then it sort of crystallizes the rest. That's
Oh,
how
that's cool.
things work as well. So if you break up the ice cube in small pieces and would throw it at different cells, they'll take it up and they'll grow big ice cubes then as well. So these things can spread in the brain.
I
Uh,
see.
the thing, it's a similar process as you probably heard of like Creutzfeldt-Jacobs disease or mad cow disease.
Hmm.
So what happened with originally mad cow disease is uh, so very bad agricultural practices, but people were feeding like pretty much carcasses of cows. They were grinding it up and putting it in the feed for cows.
Shit.
And so if one of these cows that was being grinded up to put in the feed had mad cow disease, that meant that it had like protein aggregates in its brain, like a
Oh,
specific protein that misfolded and made clumps. And so now these cows were eating these the carcass of the sick cow. And what happened is like this specific protein, that definitely probably not the case for all of these proteins, but this specific protein can like be taken up by the gut and through the nerve cells
Mm.
that connect the gut to the brain, travel up the
Oh
brain
shit.
and make the cow sick. And so that's why people then who were eating contaminated cow meat developed a similar disease, because this type of protein can actually cross the species barrier. So
Hmm.
the human version of that protein, and we call it the prime protein, uh, can be sort of seeded, like nucleated, can grow ice crystals if it sees like a small ice cube that originated from a cow. And so that is like Creutzfeldt-Jakob's disease and like mad cow disease then in cows. And even in Papua New Guinea, where people still decades ago practiced like ritual cannibalism,
Hmm.
that you had Kuru, laughing disease, they called it. So people also had this form of neurodegenerative disease, and it was mostly uh, like in like Papua New Guinea, in these tribes, if a family member would pass away, the women and children would eat the brain as sort of as like, yeah, as
Like absorb its knowledge or some
yes,
shit.
a ritual to commemorate the death.
Hmm.
And so what happened like decades later, like 40, like often these kids would eat it then, but like 40 years later, they all of a sudden would get sick and get these neurodegenerative disease. And actually they had the human equivalent of like mad cow disease, and we call it Kuru disease, where it like really spreads between individuals through eating. So the thing is, ALS, Alzheimer's, Parkinson's, all of these proteins behave in similar ways, but they don't really infect people, because like maybe just because we're not eating each other anymore, maybe it's because you lack the specific receptors to take those those specific
Oh, what
proteins
about
in the gut.
what about if my grandma, like my grandma, for example, she can't
Yeah.
move half of her face,
Uh-huh.
like so she yeah, and the way that her face folds, even like the way that her wrinkles are only on one side, right? Because
Oh
she
yeah,
can't move the other side.
she had a
So
stroke or like,
yeah, I think she might have had a stroke. Yeah.
yeah, yeah.
So yeah, I guess my question, yeah, I guess she would have had a stroke, right? Like, but my
Yeah.
question was whether this is like heritable, like if I can, I guess,
Stroke itself is not
yeah,
heritable, because stroke is like like
yeah. I was thinking she had some form of
it's
ALS, to
not
be
really
honest.
a
Forgot
disease.
that strokes were a thing.
It's like not really a disease. It's like an accident, right? There's like
Right.
a blood vessel that gets clogged or whatever, or ruptures, and like, but you can have if there's a genetic predisposition to develop
Hmm.
condition stroke, that may make you predisposed, just as like you may be predisposed to develop uh, obesity or whatever. But
But
like,
not ALS. You
I mean,
can't
with
be genetically.
ALS, the same thing. So
Okay.
there can be genetic predisposition, uh, and we are getting to more and more of the risk factors that are underlying that, and sort of it's important to know the genetics behind it, but it's also equally important to know environmental factors behind it: diet, exercise, where do you live in this world, and so on and so on. And those have been more elusive. Specifically in the case of ALS, it has been not really clear what type of environmental conditions could underlie it. What we do know is people that are playing high contact sports
Hmm.
have a predisposition. Soccer players, specifically also American football players and so on. War veterans,
That's
they have a
interesting.
predisposition for a lot of degenerative disease, Parkinson's, dementias, ALS.
Do you have
Those
a
are
theory on that?
presented in the veteran population. So, what specifically about the war environment that predisposes these people to that, we don't know yet. Uh, but it probably also, based on the analogy with contact sports, it may have to do with concussions and so on, and
Interesting.
like some brain damage earlier on in life that sort of can put you on a path towards.
Well, what about like if I get twitchy from like like from lack of hydration. Obviously that affects everyone, not war
Yeah,
veterans, but also stress, for example. So could trauma and stress like be that for war veterans, and contact sports, nerve
yeah,
damage and contact sports
yeah,
seems like a thing, right? Like,
yeah. No. So yeah, again, we have no clue. Probably like if we talk about stress, we should specify what type of stress is, like physical stress or is it
mm,
emotional. Emotional stress is obviously, mental health is important for everyone, and like having a happy outlook on life will make you feel healthier, and probably it will make you just healthier over like long time, because like it will change your eating patterns, it will change your exercise and so on. So these things are always multifactorial, right? And they can work in
of
direct
course.
but also indirect ways. And again, there is so much to explore there and things we don't really know yet.
Hmm.
uh, but yeah, like that is definitely an area of like intense study, because you sure, we're getting into the age of genome engineering, but it's still easier to change something in your environment than changing your genetic makeup. Way easier. So if you can find environmental factors that contribute to these type of diseases, like look at like a heart disease, and like look at like look watching making sure you don't have too much cholesterol in your diet. That's an environmental thing. Like what is in your diet, and will predispose me to get heart issues later on in life. That is like a success story of that, right?
Hmm.
And the thing is, it would be good to find factors like that for neurodegenerative disease as well.
Cool.
But unfortunately, those have been a bit more elusive.
Sweet, sweet. Now, in your Twitter profile, I'm gonna bring it up here. You're in a desert. Is that a desert?
Oh, that is the Oregon coast. That is the dunes of the Oregon coast.
What
And
is that?
it looks like, so here, just a few hundred miles up, like at the south of Oregon, at the coastline, you have like these large dune formations,
Oh,
which
so cool.
if you manage to get a shot where you don't see the pine trees and so on, it looks like you're in the Sahara. Like, I've been in
Yeah, it looks amazing. It looks
I've
so
been
cool.
in
Can you?
Morocco and so on, and
Uh-huh.
the color of the sand is slightly different, but the feel is exactly the same. It's like
Yeah,
you feel like you're in a desert environment. So, but that's just here in Oregon on the west coast, so not as exotic.
that's so cool. Is there sandboard?
Yeah, people do sandboard there. I
That's
have
pretty cool.
I haven't sandboarded myself, but
Yeah.
I saw people like a
What
lot
about
with.
snowboard? Have you snowboarded?
No, no, never.
You've never snowboarded? Oh my god, you're
Never,
missing
never.
out.
I only skied as a kid, but classic dad story, right? Dad was gonna learn me and my brother to ski instead of sending us to like classes, and like he wasn't the best instructor, so we
Uh,
hated it. We gave up after 2 days. My
uh,
brother later on in life picked up snowboarding, and he loved it. But
yeah, snowboarding's great.
if I have to learn now in my 20s or 30s, I'm just gonna break everything.
I don't know.
I
I don't know.
don't.
And it's a really good exercise. We've been
I
talking
know. I know.
environmental factors, man.
There's a part
Exercise and
of me
water,
that
right?
wants to do it. Yeah, but I don't want to break a limb.
Yeah.
But the
I mean,
thing is,
it's a
part of me wants to do it, but like now, I never done it, so I don't miss it. So I'm not gonna
true.
learn.
That's a good point. That's a good point. All right. Well, so what's going on with you right now? Are you still very committed to the research on ALS? What's the next thing? Are we doing mice soon or?
So I myself am not doing much mouse work. Colleagues of mine are, and sort of whenever we find something that's promising in the lab, we will eventually test it in a mouse model. Uh, I just really I try to focus in my work on like really the early steps of ALS. So I talked a lot about this protein aggregation, and sort of we believe that the a protein moving from a gas or a liquid like state into the solid is what potentially is like happening at the onset of the disease,
Mhm.
which again is probably now, as we know, decades before you really develop the symptoms and start
Mm.
dying. So if we could find some therapeutic strategies there, that would be like, yeah, be really useful, because now a lot of the therapy is aimed at trying to treat the symptoms or sort of revert the symptoms, for example, with stem cell therapy and so on. But I like, my expertise lies in sort of trying to elucidate really the molecular things that happen at the start of the disease. And so, yeah, we're still working on that. In our lab, we have done some mouse work on things that are now going to clinical trial. I wasn't involved in that work, but there is definitely like progress on the way. I'll
Awesome.
probably stay studying mostly in style models and so on. And when there are some stuff we gonna try to do in mice. Uh, but what I now specifically at this point of my career I'm doing is not take. I don't call it taking a step back. It's sort of taking a slight detour, if you will. So everybody has sort of need to find their niche in a field, right? And what are you good at? And like I realized I'm good at connecting the dots. And like I'm originally an engineer, and like what is the best engineer on this planet Earth? Like life and evolution.
Sure.
Like there's so many different engineering challenges, and life always finds a way. So what I'm now doing is taking a bit of the detour and like go study organisms that are exceptionally well at preventing proteins to misfold,
Cool.
and see if those organisms may inspire us to come up with novel strategies of how we can prevent protein aggregation in the human brain. And some of these organisms are like things like this. Like this is like a
Oh,
false
that looks.
Jericho, they call it. So this is like what they call a resurrection plant. So
Oh,
it's
what?
dried up. It's a desert plant. Uh, if you would put it in water, you can just buy these on Amazon, or like you find them in many shops. If you would put this in water, this thing will open up, will get completely hydrated, get green again, and start growing again. So this plant is officially sort of dead, but also not dead. So
That's
like it's not
amazing.
doing anything at this point, but you
It's like
can
that jellyfish, like the jellyfish
resurrect it.
that Dyson.
Yeah. And the same with plant seeds, for example. Like a plant seed is literally a plant embryo that is completely dried out and just sitting there being dormant. Imagine taking a mouse or a human embryo, just completely drying it up and then
Yeah.
pouring water on it and hoping that it will live again. Yeah, that's not
Yeah.
working.
I mean, there's a story or two of people being brought back, after a couple of hours being dead, but these
Yes.
plants will
Yeah.
probably stay like
Yes.
seasons dead, right?
And so the thing is, what proteins need to not aggregate, like they always need, like, if you remove water from around the protein, it will start aggregating.
Hmm.
Take a big bit of egg white from an egg, just put it on your kitchen counter and let it dry out. You'll get this like dried out film. If you then pour water on it, it's not, doesn't immediately get the same texture as egg white, like, because the protein's literally aggregated, right? And sort of, we still don't fully understand why plant seeds, these resurrection plants, for example, tardigrades, these things they ship up into space and are super resistant against any type of
Oh,
stress.
those are the gross caterpillar-looking things.
Yeah.
Yeah,
A
tiny,
friend of,
tiny.
you should have him over. Like a friend of mine is actually studying these things.
Really? That's so
Oh,
cool.
yeah, he's amazing. So we still don't fully understand why these organisms are so resistant against protein aggregation. Because obviously, if they would dry out and their proteins would aggregate, and you give them water again, they wouldn't be able to survive because all of their cells would be full of these toxic protein aggregates. So these organisms, over millions of years of evolution, have found a way to prevent protein aggregation. And the type of proteins that aggregate in our brains are actually also found in these type of organisms. So proteins that look very similar. And so in collaboration with the relab here at Carnegie, like we have been studying some of these proteins that look like the ALS proteins, but in plant seeds. And we see that they function in surprising ways, are actually super helpful for normal physiology of a plant. That is like switch between gas and liquid is super important for their function. And so what we're now trying to do is figure out, even though the plant seed dries out, why doesn't this protein irreversibly aggregate? How is the plant seed preventing that? Because if it can do it for a plant protein that looks like the proteins that clump in our brain, then maybe we can even like trick these proteins to not do that in our brain as well.
Mm.
And again, I don't call it taking a step back from ALS research. I think it's sort of, I describe more as taking a detour and see what's out there and sort of be inspired by how evolution has come up with solutions to the problem we're trying to solve in ALS. And
Yeah,
in the,
that makes perfect sense to me.
yeah, and in the meantime, what was actually very interesting, and like initially I didn't think about this, uh, but so by discovering now a protein that sort of can uh, function like that in a plant seed, like switch between gas and liquid state, we actually found a protein that can we can control when a seed will germinate. So normally a seed will germinate if it gets a certain amount of water, because germination is a risky thing. Like the moment the seeds as a seed, like the seed is very stress tolerant, but the moment it germinates and you have a small seedling, if it doesn't rain the day afterwards and there's not enough water, the
It's
seedling
done.
may die
Mm.
because it's not stress tolerant anymore. So the timing of germination is really a key thing plant seeds need to control. And sort of by engineering the protein we discovered, we could trick plant seeds into thinking there wasn't enough water or there was enough water when there actually wasn't. And so we could
Oh,
really time their response to water for germination. And so why this is important, like obviously climate change is a big problem ahead of us.
mhm.
And drought is gonna be a thing. And so our agriculture will need to adapt. And what is interesting is like that currently one of the big problems farmers face is when they sow their seeds, they can't control the timing of when the seeds will germinate. When it's like exactly when it rained, when they threw in fertilizer. Some seeds may immediately germinate, some will wait a day, some will wait a week, and some will never germinate. And there's an entire industry trying to make seeds germinate as soon as like prime. They call it priming seeds. They will try to mechanically like treat the seeds so they will germinate the moment they are thrown on the field. But nobody ever figured out a real like strong genetic factor that did this. And now this by studying a protein because we were interested in it from a brain disease angle, we now were able to engineer plant seeds that we can really control when they would germinate and so on. And so we think it's like this cross pollination between different fields that is this really gonna be instrumental in bringing ideas from between different fields and like really start engineering like some of the problems that are ahead of us, whether that is like aging and neurodegenerative disease, whether those are like infectious disease like viral infectious and the post antibiotic era, or whether this is climate change and the collapse of our agricultural system. I think there again patterns. It's all about
Yeah,
patterns and conditions. There are solutions to the problems we have in a specific field. People in another
somewhere
field
else. Yeah,
already solved it, or an organism has already solved it. The
yeah.
only thing we need to do is identify that and see how we can re-engineer that process or re-engineer that organism into solution for the problem we face in our field.
That's incredible. And I imagine, well, we saw your Twitter profile in the desert. I imagine that desert life is particularly good at this in general, right? Like,
Yes. Yep. No.
yeah.
And like there is so like life, like every pretty much everywhere on earth you'll have some form of life. So
Mhm.
life is extremely resilient, and what we think as an extreme environment is, yeah, for some organisms just a chill
Nothing.
sunny
Yeah.
day in California.
Yeah.
Living in a boiling acidic like geyser in Yellowstone is like the same as me lying on my hammock here on the patio.
Yeah.
For some organisms, for us, it would be pretty lethal, but
Like the tardigrades, the tardigrades,
yeah,
those fuckers, they can be at like almost any temperature, and yeah.
they are phenomenal. Like, and again, that the cool thing about them is like they do the same trick as plant seeds do. What they do to become stress tolerant is they desiccate. So they lose all of their water
They
content, they
die.
shrivel up, but in the process of shriveling up and drying out, uh, they produce a lot of proteins that will prevent the rest of their cellular proteins and in cytoplasm to aggregate. So they have sort of these proteins that keep everything in place and prevent things from aggregating. And that is why they're so stress resistant. And I'm not gonna say anything more about tardigrades. You just have to get Tom to be here.
All right, you'll have to
he's,
put me in
he,
touch then.
he's the true rock star.
That's awesome. Yeah. All right. So that's amazing. So that's what you're doing right now is looking into I
Yeah.
guess organisms and
Mhm.
ideas in
Yeah.
the
We
agricultural
still
field.
have a bunch of ALS projects always in the works. Like we always keep on pushing the ALS angle. But I have a bunch of side projects where we like, yeah, try to do wacky things and like
I
just
think that's it. Yeah.
completely look for a pattern and then and pursue it. And it has led me to study plant seeds. It has led me to study bacteria. Now studying rattlesnake venom. Like there's a lot
Rattlesnake venom.
of exploring and
Is it because it's paralyzing? Is there some threat there?
yeah, so rattlesnake venom is a mix of small molecules and small proteins and so that are evolved to kill a prey, right?
Mm-hmm.
And so what we figured out is that some of these small proteins that are found in rattlesnake venom actually look like some of the proteins that are found in certain types of ALS patients.
Oh shit! So they inject the jam and they paralyze you type thing with it,
Yeah,
or
so like they inject and there's like a cocktail that will make some cells just die, some cells just paralyze and so on. But we see like sort of we were studying a specific type of protein found in certain familial forms of ALS, so people with a specific mutation. And only these people have these type of proteins. And again, they're repeat proteins. So again, repeats haunt me everywhere. Uh, and so I had been studying these type of proteins for the last 7 years or so, and just was thinking like, okay, I'm gonna look at other organisms and see if maybe these type of proteins actually exist somewhere in nature. And yeah, they do, as antimicrobial proteins, as venom proteins, and so even our body sometimes produces these types of proteins. But and that's the thing, regulation is key.
hmm,
If you produce
balance?
protein in the wrong amount, at the wrong place, the wrong time, you get sick. If you like, it's playing with fire, but if you have it under control, it can be a really good instrument if you want to kill something.
Mm.
So our body uses these type of small proteins to kill bacteria. Uh, Rattlesnakes use those type of proteins in a cocktail of a lot of proteins to kill a prey. And again, if it I could, it's exactly, I think the comparison with fire is a really good one. It's pretty dangerous if it goes out of control, but if you have it regulated, if you have it contained, it's like one of the best tools you can have, right? Like
Yep.
look at humans. Where would we be without the discovery of fire? Like,
Yeah.
and so again, yeah, I think there is like nothing in nature is unique. A lot of things in a way, shape, or form will be found somewhere else. Specifically, if it's like a powerful concept or a powerful mechanism.
Hmm.
Evolution has had billions of years to play around with things and look for the best solution, and often like the best solution is already somewhere out there, and evolution will
Yeah.
stumble again on it. Look at like convergent evolution, right? Like the development of wings in like birds, in like bats. They're both wings, but they like evolved evolve differently, but they come to the same solution to the problem.
What's
And I
with wings in chickens? What do they have wings for? That's what I wanna
like that,
ask evolution.
like chickens can still they can still fly, right?
Yeah, they can kind of fly a little bit.
Yeah, the thing is what we do with our domesticated chickens is like we often actually, and I know this like growing up in a small town in Belgium, we always had like yeah, chickens, ducks, rabbits, like sheep and
Nice.
whatever. But like what you do initially with like small chicks is sort of you cut a bit of the wingtip, and
Oh,
so
I see.
then they're like, if they want to, like they don't grow like these specific feathers on the wing that will like really give them the lift.
I see. I
You
see.
do that on one side, and then they're sort of in balance, and then that's how they can fly. That's why domestic chickens don't really fly. But like if you're gonna look at the wild chickens, like the jungle fowl in the jungle of
I didn't
Southeast
even
Asia,
know there
has
were
to fly
still.
away for tigers, right?
Or I
So
guess, yeah.
they just fly up in the tree. They don't
Really,
fly.
up in the tree, that high up, like just fly up.
No, they can fly in a tree, right?
Oh wow. Okay.
Yeah, and no. They don't fly between trees, right? But they can like they jump and they fly up, right? These
Yeah,
things
yeah.
can.
I've seen chickens jump high and then kind of like
Yeah,
they kind of
yeah.
kind of
Our
like
domestic chickens,
Mario Bros.
yeah.
Yeah,
Our domestic chickens are also like, yeah, they're not really selected anymore. They're
yeah,
selected for meat production often, so they're like, yeah, super bulky and whatever. I don't think
yeah.
they're
That's a good
to
point.
fly.
That's so funny.
Yeah, chickens and wings.
Yeah, good times. Well, I've completely loved this. I hope that we can meet soon and maybe do a live episode or talk to you again
No,
at some other
yes,
point.
I should be in the coming months, maybe in Irvine. So
Hit me up for sure. Yeah, I'd love to grab a beer. And yeah, we'll keep in touch, I'm sure. Do you have any final shout outs or even any
oh,
other topics? Because I've just run out of ideas, but if you have anything else that you'd like to talk about
oh,
too.
yeah, no, like I think like I definitely have a shout out to Alan, right? Because
Hells
like
yeah.
because you guys were friends, like that's how we got to know each other. So and like Alan has always been a bit of a personal hero, like
He's great.
great scientist, great artist, very fun to hang out with. So like yeah, it's
Absolutely, yeah.
it's cool that's how we got in touch.
Indeed. Yeah. Much love to Alan as well. If you guys want to check out Alan, that
Drummond.
was Alan Drummond. That was the first episode of my podcast, and we'll
Yeah,
continue to do
no.
these. So thank you very much, Steven, for coming and talking to us.
No,
And
this was amazing. Yeah, and I'll hope to see you guys soon. And yeah, thanks for having me.
alrighty, take care.